what we see crossing the blood-brain barrier and really getting into the tissues, that's where we want these therapeutics to go, or is it really just exosomes circulating around the brain in the vasculature? So we partnered with a person at UT Southwestern, Denise Ramirez, and analyzed our brains, our mouse brains, by serial 2-photon tomography. Those are the panels on the right. And what Denise showed with our exosomes and our tissues is, yeah, these exosomes really penetrate or get across the blood-brain barrier and accumulate in various regions of the brain, including hippocampus, cerebellum, cortex, and a few other regions. So they do get across these barriers. A big question also is, can we load these milk exosomes with therapeutics? I mean, because that's what we ultimately have to do, and I'm going to show you a few examples later on. I mean, so this is just a space holder right now. Another feature that's very important, if this ever was used in human patients, is these milk exosomes are apparently safe. So we did a few studies in humans, did not observe any cytokine response, and we did also do studies in mice showing, okay, liver and kidney function were normal after milk exosomes were administered for a long time. No immune response, food and water intake was normal, physical activity was normal. So no worries yet, at least, about cytotoxic or, well, safety concerns. So all this is really exciting, but of course, like with everything in life, there's limitations to nanomedicine, and these are 3 of the major limitations that you encounter when working in the nanomedicine field. Many of these nanoparticles, including milk exosomes, are rapidly eliminated by macrophages, immune cells. So this is not what you want to happen, because all the therapy and therapeutics would be eliminated and erased. And then the other problem would be tissue homing. How can you make sure that your therapy or your treatment goes specifically to the tissue that you want it to go, as opposed to go everywhere in the body and elicit adverse side effects? And then so those are the 2 that I'm going to show you some data about. And number 3 would be if exosomes or nanoparticles are taken up by cells, they're also rapidly degraded by a particular compartment in a cell called lysosomes, and that means about 80% of the cargo that you deliver is degraded in cells. So this is also something that you have to overcome if you want to make this a really successful or perfect system. As a workhorse right now, we use what's called bovine mammary alveolar MAC T cells, and we use these cells because it's easy and fast to work with them when making genetically engineered exosomes. The other reasons are they have morphologies, especially at the bottom, very, very similar or almost identical to cow's milk exosomes. They have sizes identical to cow's milk exosomes, that's the red and orange on the right. They are orally bioavailable, just like milk exosomes, and they have tissue distribution patterns also. Okay, also, I thought you are. Tissue distribution patterns, I can step a little closer. Tissue distribution patterns very, very similar to normal milk exosomes. So let's give you one example of the modification that we did to our milk exosomes in these MAC T cells. Remember I told you about the elimination of these milk exosomes by macrophages being a big problem. So there is what's called don't eat me peptides. They're called CD24 or CD47. So we engineered our MAC T cells to secrete exosomes that were decorated with either CD24 or CD47. The left panel shows you that these decorated exosomes are taken up less efficiently by macrophages in cell cultures, which is what you hope for. So about 50% decrease, and this was even more impressive when we did this whole story in mouse models in a whole organism. When you see after 2 hours or 2 hours after tail vein injection of our exosomes, the plasma levels of the CD47 positive exosomes were about 12 times higher than the plasma levels of the non-modified exosomes. And this slow elimination of milk exosomes, of modified exosomes, was associated with an increased accumulation of milk exosomes in the brain. As you can see, this bright red brain on the left, this is the CD47 positive exosome. The middle panel is the CD47 negative exosome, still gets to the brain, but obviously much less efficiently. And left, the right, which I call PBS, saline, is just our vehicle control. And this increased accumulation in the brain also benefits the delivery of CD47, of SYNGAP gene to the brains, that you see on the right. So the red bar, tiny, is just untreated control expression. The middle bar, the yellow, is CD47 negative exosomes delivering SYNGAP to the brain, which is quite effective. But then you can see an even more effective delivery if you use CD47 decorated milk exosomes. So I know this is not a cancer crowd or audience, but I use an example from our work in the brain cancer field, to illustrate the power of these homing signals attached to milk exosomes. We partnered again with our friends at UT Southwestern. They're brain cancer people, and what you see on the left side is brain tumors, human brain tumors growing in a mouse model. I mean, a mouse xenograft model of glioblastoma. So this red that you see is the brain tumor. Then the middle one in the green, this is our milk exosomes decorated now with tumor homing signals, COOP and ApoE. And you can see, or you can already imagine, apparently these tumor homing signals take the exosomes to the brain tumor. And then what you see on the right side, the yellow, is the merged images. So basically telling you there's the perfect co-localization of milk exosomes decorated with these tumor homing signals in the brain tumor. And this collaborator of ours, he's an established brain cancer person, told us he has never seen any localization as perfect as this. And this speaks to the technology also working with a specific homing specificity of genes in the SYNGAP field. Exosomes, you want to be sure that these cargoes that you put in there, whether it's genes or siRNA, are biologically active. And so what I'm going to show you here is a regular milk exosome loaded with a plasmid coding for a fluorescent protein. And the test here is basically showing that these cargoes, the plasmid, can get out of the exosome into the cytosol and be expressed in the cells in the brain. And what you can see on the left side is the very far left mouse is a mouse that received this fluorescent protein, but without densitometry analysis. The mouse right to it is the mouse that received empty exosomes, so basically no plasmid. And you can already tell here there's definitely a signal difference between the two. And then on the right side, we did some densitometry analysis. So now you can see the treated mouse with this red signal in the brain compared to the untreated mouse to the right of it. So you have seen this slide before at the very beginning. So this is the status quo. That's what we have right now. We are playing with doses and everything. But what I was going to show you in the last, I hope I have a few minutes, Mike.
2 minutes.
Oh, perfect. Yeah. So we want to do some position us for gene editing. So we actually introduced Cas9 in our milk exosomes, and this is important. We're almost there in our pursuit of developing what we call a universal milk exosome. So this universal milk exosome is a particle that can go anywhere in the body where you want it to go, but only to that particular place. It can also deliver anything that you want to deliver to this particular location or gene or region of the body, and does so with great specificity. And I remember about a year ago, I think SRF invited me to give a webinar, and some of you may have attended. Back then I was talking about goals, and I cannot tell you anything about top secret, and now we submitted the patent, so I can talk a little more about this, so I can take the black box out of the game and show you what we have in mind for our universal exosome. So this is what you see on the left side now. There's the Cas9 that is in there, so we could actually load the exosome with guide RNA to actually edit genes. And as you can see, there's 3 membrane proteins, CD9, CD81, and CD63. So we are using a combination of biology and chemistry, specifically bioorthogonal click chemistry, to modify CD81, which is this membrane protein. And we do replace 3 natural amino acids in this protein with unnatural amino acids that have A site, alkoxyamine, and alkene moieties. And I'll tell you in a minute why this is important. And the CD81 also has a polyhistidine tag shown in red, which allows us to purify these milk exosomes with great purity by very economical nickel 2+ resin, and then use these very pure milk exosomes, which eliminates some safety concerns, and attach what you see here is a triangle, pentagon, and I don't know what this other thing is. Attach our peptides that we want there to be. So for example, we could attach a homing peptide, like I showed you for the COOP and the ApoE in the brain, or in your case it would be RVG to get these exosomes to the brain and nowhere else. We could attach this blue pentagon, a don't eat me peptide, let's say CD47, and we could also attach, or will attach, a retrofusion peptide, which eliminates or reduces the degradation of these exosomes in lysosomes. And I think that's your conclusions. So milk exosomes have qualities conducive to the delivery of therapeutics, and that obviously includes SYNGAP genes. MAC T cells are a useful screening tool for our lab purposes, and then transgenic livestock, so the biofarming, secreting modified universal milk exosomes seems to be a promising technology. I think the people in my lab, the funding agency, also want to call out Hans Schlecht back there in the room, the mask. He was the one reaching out to me in the first place, and I think we have developed over the past 1.5 years a very friendly relationship, and I single you out on behalf of all the other SRF leaders and SRF community. I think this has been a great journey for me, and I didn't know anything about SYNGAP literally when you emailed me 2 years ago. I thought, oh, this is something I need to read up quickly, and this has been great. It's a great experience, and with that, well, there's probably no time for questions, but okay.